Yeah, that's right. Okay. Hi, everyone. Sebastian, I'll take Sebastian's place as well here at the moment, and just remind you to go ahead and turn off or silence your cell phones. So it's my pleasure to welcome you to the final Friday evening lecture of the 2019 season. My name is Nipan Patel. I'm the current director of the MBL. I regret that Marty Chalfie couldn't be here this evening. There was a medical emergency in his family. But tonight, it's a real pleasure to dedicate this Friday evening lecture to the memory of Osamu Shimomura, a former distinguished scientist at the MBL and a 2008 Nobel laureate in chemistry. We're honored to have amongst many of the guests today, many of his close colleagues and friends, and especially his children, Sashi Shimomura and Susuma Shimomura, and their mother, Akemi, sends her warmest greetings from Japan. So Osamu Shimomura was a senior scientist at the MBL from 1982 until his retirement in 2001. And he dedicated his career to the study of bioluminescence in many different organisms, particularly trying to understand the chemical structure of the photoproteins that created that bioluminescence. And in 1961 to 62, he discovered and isolated the green fluorescent protein, or what you might hear commonly referred to as GFP, which has been a revolutionary molecule in biomedical science. And in the 70s, he was also able to elucidate the structure of the chromophore and understand exactly why it is that it's able to fluoresce the way that it does. And then he was awarded the Nobel Prize in Chemistry in 2008 with co-recipients Marty Chalfie from Columbia University and Roger Chen from the University of California, San Diego. So I'm pleased to introduce tonight's lecturer, who is a friend and a colleague of Dr. Shimomura, and has been instrumental in the study of bioluminescence in many different contexts. So Steve Haddock is a senior lecturer and senior scientist at the Monterey Bay Aquarium Research Institute, and also holds an appointment at the University of California, Santa Cruz. So Dr. Haddock studies marine diversity, including molecular biology and bioluminescence, and in all manner of organisms that live in the deep sea and at different depths in the ocean. And in addition to conducting research expeditions to go after these bioluminescent organisms, he also is very much interested in elucidating and understanding the relationship between different organisms and in understanding the vast variety of proteins that are used to create light. So he received his BS from Harvey Mudd College and a PhD from the University of California, Santa Barbara. And so it's my pleasure to introduce Stephen Haddock. Thank you very much.
Good evening. In keeping with the theme of tonight, we're going to dim these lights down a little bit, but don't fall asleep quite yet. I wanted to just thank Marty Chalfie, who was willing to come here and introduce me, but he couldn't be here tonight, as was mentioned. So I think he would probably want to make these 4 points in his introductory remarks, so I'll just let him make them there for you. I completely endorse all these messages. Oh, as well, before we get started, I should clarify, since we are in New England, that my name is Haddock and I am a marine biologist. Nobody in California even knows what a haddock is, but out here I just get, sideways looks. So I was really honored to be invited here to be able to speak in the context of the life of Osamu Shimomura. I just found out at dinner that today is actually August 9th. It's the anniversary of Nagasaki, and he was actually blinded by the bomb at Nagasaki. He was watching the plane fly in, and he was blinded for temporary. So I'm I don't really know how to treat that information, but anyway, I just realized that or learned that. So I first, I think, met him at a scientific conference about bioluminescence, and then every time I was in Woods Hole working with other people, going on a cruise or whatever, I would just randomly pop into his lab unannounced and say, how's it going? And basically pick his brain and get his wisdom. And he and his wife were just incredibly gracious and hospitable to me. And I learned a lot just being there, conversing with him and strategizing about experiments and how to do science, I think, properly. He had a lot of really good ideas about that I try to pass on to my students as well. There were also some more tangible benefits to being an associate of his, and one is that he actually sent me some purified cylindrazine, which he sent in this little tube, which I still have. This is just a mock-up of it, obviously. But to me, this was sort of like, the amulet that in Lord of the Rings that has like glowing light, and it was just like this holy object. So he sent it to me when I was a grad student. I unpacked it. It came in this little tube. I got in the fume hood. I got out, my pipette. I was supposed to resuspend it in methanol. And so I squirted the methanol in there, and it went shooting right back out because I thought that whole tube was the tube, and the bottom part of that was glass, and the little reservoir at the top was the part where the cylindrazine was. So I was mortified, but fortunately it was so concentrated that I still had plenty that I used to do experiments for years after that. And another colleague who later sent me some cylindrazine, he in his little cover letter, Bruce Bryan, he said this is about $500,000 worth of cylindrazine in market value in this tube. So it's gotten a lot cheaper since then, but this was really a treasured gift that he gave to me, and it actually enabled a bunch of my early work. So all these papers directly used that cylindrazine that he had given me, and most of them end in in some kind of an acknowledgement like this, where just to appreciate. So his fingerprints, really, you'll see some fingerprints around here in the corners. They're in a lot of the work that I was able to do. Another thing, there's a kind of a legend if you're a bioluminescence researcher, that during the war, soldiers in Japan, this is one of those supposedly things, would have these little sesame seed size shrimp that they would rub on their hands and use it like a flashlight when it would bioluminesce in their hands. So they would get it wet, spit on your hand, crush it, rub it up. And so when I went into his lab for one of these first visits, there up on the shelf is this big jar looking suspiciously full of sesame seed shaped things, and it said Japan 1945 on the side of it. And I was just blown away because this was like captured, moment of history. And it turned out that was something that he had gotten. I guess they had been drying these. They had collected them by the bazillions and dried them out on these black tarps, and then the war ended, and so they didn't have use, but they scooped them up, and he had these in his lab. So he gave me a packet of those, which we're able to photograph here. This is a photo by David Lichtwacker that we did in my lab using some of the ostracods that he had given me. And as you'll see later, that was where he first got his real break in bioluminescence. Another one of his influences was this book that he wrote about bioluminescence. And I was fortunate enough to be able to provide the picture that he used on the cover, and also to do copy editing for the English inside of it. But in the appendix, and it's funny because this has come up a couple times today with my students and also with his son, but this is practically the very last page in the book is a section on advice to students. And it should be required reading, I think, for all scientists, even not just for bioluminescence. Another thing about the book, in the first edition, I wrote the little blurb on the back cover, and I basically said that, having a book by Shimomura about finding bioluminescence chemicals is like having a book by Humphrey Davy about finding elements. And you may think this is hyperbole, but if we think about it, so Humphrey Davy discovered and characterized all these elements that, across the periodic table. Well, Shimomura studied all of these organisms and made fundamental contributions to our understanding of bioluminescence and the biochemistry, especially, of bioluminescence in all these fields. So he really was like the Humphrey Davy of bioluminescence chemistry. So instead of launching into a bit about his research, I was going to give you some background about some of the concepts that are going to come up to be important later. And so these are the four main things that I want you to take home. This used to be titled the quiz, pre-quiz, post-quiz vocabulary, but for tonight, it's the background. So I'm going to talk a little bit about ctenophores and siphonophores because I think most people are not as familiar with those, and then also try to disentangle this idea of fluorescence and bioluminescence. So I do research in California. We're just below San Francisco, adjacent to the Monterey Bay. This place called the Monterey Bay Aquarium Research Institute. If you come visit, we are not the Monterey Bay Aquarium. You'll have a 30-minute drive to come up there. So we're relatively small. There's maybe 250 people work there, and we focus on studying the deep sea largely. And actually, my decision to go here was also influenced by Professor Shimomura, because at the time I had a job offer to go to a university and teach. And so I asked him, what do you think I should do? Should I go to this little research institute or should I go to teaching? And as you might predict, he said, go do the research. You can always teach later, like later in life when you're retired. So that helped solidify my decision to go there. So at our institute, we use some different techniques. One of them is what we call blue water diving. This is scuba diving, but it's more almost like a spacewalk where you're just in the three-dimensional environment. The bottom could be 3,000 meters below you. It often is. And you're connected by this series of lines that go back to a little float, but you're basically launching off a small boat and collecting things by hand into small glass jars. And actually one of the pioneers, Larry Maiden, was one of the pioneers of this method, and he's here tonight as well. So that gets you into the upper 20 meters. To go down deeper and collect things, we use remotely operated vehicles and sometimes manned submersibles. And these let you be on the ship together, and it's almost like doing a video game where you've got your joystick and you're flying around and try to get a high score of collecting, the smallest, most transparent jelly that you can find. And we basically drive them into these samplers and then close the sampler, and it lets us get organisms intact that is really important for studying bioluminescence, because if you drag them up with the trawl net, they've already done all the flashing that they're going to be, able to do by stimulation. If we collect them this way, we can get them in really, really good shape. And so with these methods, we study a lot of what are called gelatinous organisms. These are not related to each other really closely, but they all live in the open ocean or in the water column, so they're all transparent and relatively fragile. And then tonight, again, I'm going to talk about these two guys in particular. So first for hydrozoans. So this is the Medusa aquaria that Dr. Shimomura did a lot of his work on. And this is a type of Medusa that has alternating life cycles. So not to get too technical, but there's a little part that sticks up off the bottom. And they have subdivision of the labor, essentially, where some parts are for feeding, but they can't reproduce. Other parts are reproduction, but they can't feed. So one part will eat it, distribute that digested material to the rest of the organism. But we have these things called siphonophores, which are related to those hydromedusae. And these, I think, are pretty confusing to most people because they're said to be colonial. And so you hear people say, well, they come together to form this one organism, but it's really not like that. If you think of an analogy with those hydromedusae I just showed you, what if they don't have a substrate? What if there's no bottom for them to attach their polyp to? So that polyp is just sort of floating around in the water column. Well, in the case of siphonophores, they usually add some swimming bells for pulling that whole colony through the water, but they still have this subdivision of labor where there's feeding polyps and reproductive polyps that trail along behind. And the feeding polyps have a stinging tentacle that they use to catch prey. So I like to think of siphonophores almost like pelagic hydroids because they're similar in a lot of ways to those hydroids. So they can be extremely long. This would stretch from me pretty much to the back of the auditorium. This has two swimming bells that are about 6 inches long, and then the rest of the colony, each one of those stomachs has its own tentacle, and each one can independently catch a shrimp. So you can see that one pulsing along. Those parts that are pulsing, they can't feed. This is one I'll talk about later that eats fish mainly. And so that top part is all the swimming parts. And again, they have to rely on this bottom part to catch something, feed it, and send it up through that tube. And that tube, that's where they get their name, siphonophores. So the next group that I'm just going to whiz by, each one of these is an hour lecture of its own, but are the ctenophores, the comb jellies. And these are named for these paddles that they use they use them in a lot of different ways, mostly to move themselves through the water. This one, Baroe, Sid Tam here works a lot on this, and they actually use their cilia as teeth to bite off bits of prey. They use them as sensory for telling their orientation. And this one has these extraciliary parts that it uses to startle prey so that it jumps into the lobes instead of swimming past when they feed. So again, these will motor along and sometimes here, but that they make rainbow bioluminescence, that's not bioluminescence. We haven't gotten there yet. They use sticky tentacles instead of stinging tentacles when they do have tentacles to catch their prey. And they come in a lot of different shapes and sizes. And importantly for tonight, they're pretty much all bioluminescent except for a couple species. So again, that was a paper that was enabled by contributions by Dr. Shimomura, where I was able to actually assay the ctenophores with cylindrazine to show that they couldn't make light when they had cylindrazine. So let's talk a little bit about the essences. A lot of times I'll give a talk, and at the end someone will ask a question, and they'll use fluorescence where they meant bioluminescence. So I try to just hit that right up front and explain a little bit the distinctions between these two interrelated concepts. What makes it extra confusing is that some people like to use this term biofluorescence. I would just steer clear of that. You see it in the media all the time, but you can see how that would be even more confusing enough. It's already confusing enough. So the way you tell bioluminescence from fluorescence is by turning out the lights. Do you see any light? If yes, then that's bioluminescence. If you don't see any light, that's fluorescence. If you shine a blue light or a violet light or a short wavelength light on it and you get light out, then that is fluorescence. It only lasts as long as you're shining that excitation light on it. And I'll talk a little bit more in detail about it. Now, what gets extra confusing is that sometimes bioluminescence is the light source for fluorescence, okay? And this is the case in the jelly that Dr. Shimomura worked on, where they have a bioluminescent and fluorescent system, and one makes blue light, but it gets transferred to green light by fluorescence. Anyway, so those are the two concepts that we will dive in a little bit more into detail when we talk about the research. So how does bioluminescence happen? It's a chemical reaction that releases energy, and that energy comes out as a little parcel of light. And in bioluminescence system, there's this molecule called a luciferin. That's just a generic term. It doesn't mean any particular chemical. The firefly luciferin is different from the jellyfish luciferin. There's also oxygen involved, and then there's some kind of a protein to make those two get along and actually react. So that's called the luciferase. So the luciferase provides a little cage where the oxygen and the luciferin can get together, and when they do, boom, you get your light out, and the luciferin gets consumed and the oxygen gets turned into CO2. So you end up with oxyluciferin and, of course, your friendly photon that was emitted. So the way organisms use this luciferase is actually they can control that interaction between the luciferase and the light-emitting substrate, the luciferin. And the way that looks in real life is like this. So this is a jelly called Atolla that has a luciferase, and it gets into this frenzy where if you stimulate it enough times, it'll just keep pinwheeling out its bioluminescence. And so this is a luciferin-luciferase reaction that is happening, those two chemicals mixing. So what I'm going to try now has never been tried before. Do we have any volunteers? How about you? Do you want to volunteer to help me? Are your clothes expendable? So hold this towel like that. Actually, here, turn sideways like that. There you go. Okay. And then can you take the lights down a little bit more, the spotlights? This one up here. Don't have control over spotlight. Here it comes. All right, it still might be a little bright, but we'll try it. So this is a squirt gun, but it's a special squirt gun that Bruce Bryan, he started a company called BioToy. It has luciferin in one side and luciferase in the other. So let's see if it, oh. So you can sort of see it on the towel, I guess. Anybody want to get sprayed? Just kidding. So you can see inside the nozzle. Thank you, good job. Let's try it down here, maybe darker. You can see inside the nozzle how they're mixing right when they combine. And this is actually, so it's freeze-dried luciferin from a jellyfish, which Dr. Shimomura discovered, and freeze-dried luciferase from this copepod called Gaussia. So actually some organisms have that, almost that identical mechanism where they have two nozzles and they shoot out the chemicals into the water and leave a little puff of light in front of them. So that's luciferin luciferase, and that was the basis of bioluminescence before Dr. Shimomura discovered another one, which is these photoproteins. And in photoproteins, effectively the luciferin and the luciferase are already bound together with their oxygen, and that's in a stable thing, stable forms. And it doesn't release light until it binds calcium. So that gives the organisms another way to trigger light output by putting calcium into a system that already has photoprotein in there. So here's our friend Boroi, the ctenophore, and then again, here's the bioluminescence that Boroi makes using those photoproteins. Okay, so those are some of the principles that in the context that Dr. Shimomura was working under. So again, when he was in Japan, he started studying at the School of Pharmacy, and these are some drawings from his notebooks that were from some slides that he made. You can see just how intricate and beautiful these are. His project was to try to isolate and characterize the light-emitting luciferin, again, that little light-emitting molecule, from the sea fireflies. So the same one that they rubbed on their hands that I showed you that picture of. And he actually succeeded. I think there was a bit of chance in this where he left it out accidentally overnight. I'm not totally positive on that story, but he came back the next morning and these beautiful crystals had formed that allowed him to get the structure of that light emitter for the first time. And the researcher Frank Johnson heard about this, so he invited Dr. Shimomura to come join him in Princeton. So he left Japan. This must have been quite an emotional day to get on a big ship and sail across to the lab of Frank Johnson. And essentially no sooner did he get there than the next summer they set out in the station wagon to drive across the country with Frank Johnson at the wheel, and presumably he took this picture as they drove from New Jersey across to Friday Harbor, Washington, where these jellies, Aquaria, can be collected in large numbers. So again, this is this Medusa that was the subject of all this hoopla. It produces this bioluminescence in a little ring of light. And again, you'll sometimes see pictures where it looks like the whole bell is glowing, but really it was just that ring of light. So they would go to Friday Harbor each summer and collect jellies with these fancy samplers, the remote jelly grabber. We actually have two of the jelly grabbers in the audience tonight. You'll notice that Sashi has the beginner stick, but Stomu has graduated to the adult stick at this point. He said that the children were 3 or 4 years old when he put them to work to collect jellyfish. And then they would use the short nets until they got to about 8, and then they would get to use the big nets. So they would scoop jellies out of the water. We actually, this summer, we got to take my 3-year-old to Friday Harbor. He was so excited about it. So we live in Santa Cruz. He's like, Washington's better than Santa Cruz because I get to collect jellyfish there. We arrived at midnight, our ferry was late, and he's like, he would not go to sleep until we went down to the docks and collected jellyfish. So it was really, really fun. So they would collect jellyfish, and then they would try to isolate the tissue from around the margin. And initially they were hand cutting these things with scissors, but eventually they kind of iterated through this machinery, which was the jellyfish slicer that allowed them to process many, many jellyfish per day. Within a summer, he said that they would get 4,000 jellyfish, two and a half tons of jellyfish in a summer. And over the course of the research, 850,000 jellies were collected. So a summer's worth of jellyfish would maybe give you 150 to 200 milligrams of protein. And then that would be what they had to work with to try to figure out, the mysteries of these chemicals. At the time, they were not known. They banged their head against the lab counter trying to figure out a luciferin-luciferase reaction. And then also, I was told, but it's not in the book, that he said that he was just frustrated one day. He poured the chemical into the sink and got a flash of light out just when it hit the sink because it had some salt water, some seawater in there, which had calcium in it. And they hadn't even, this was a totally new kind of chemical reaction than the luciferin-luciferase that they had been looking for. I want to make a point. So Dr. Shimomura was, I wouldn't say anti-genetics, but he thought it was like for lazy people and it was a total shortcut, which I totally agree. I do it because I'm not smart enough to do the chemistry that he was able to do. But one thing that the advantage is that we can collect one jellyfish with our submarine, we can sequence the transcriptome, and now we can know what the photoprotein is. What the photoprotein of this deep sea organism that may not even have been described yet is. So there's a lot of, you still need to have the people who are able to do the hands-on chemistry when you're trying to solve an initial system. But once you have a bit of a foothold, then you can leverage genetic techniques to do quite a bit. So in the course of the research, there were really three pretty fundamental things that that group discovered using the jelly. So this is a diagram showing the color of light that comes out. So again, this has a photoprotein, which would make blue light. If you add calcium, you get blue light out. But it also has this green fluorescent protein that at the time, it was kind of just a byproduct that would come out as they were looking at the photoprotein. And in the case of the jelly, that sucks all the energy out that would normally come out as blue light, and it comes out as green light. So they figured out the photoprotein and the green fluorescent protein, and then also the structure of the luciferin, the light emitter that is used in that thing. And so these are just like, really foundational discoveries in the world of bioluminescence. It's kind of funny that the one that got all the attention is the GFP, which wasn't really his passion, I think, and main focus. But as Stomu said earlier, he wanted him to consider it as a Lifetime Achievement Award. And it wasn't just about the GFP, but it was more like his body of work. I should point out that there were a lot of other people involved in the GFP work. And one interesting story is this guy, Doug Prasher. You'll notice he's the first author on both these papers where they first cloned the GFP and first cloned Aquarium, calcium binding protein. This guy, it's like, wow, he must be really famous and running a huge lab right now. He's actually a car dealership shuttle bus driver. He was at the time that the Nobel Prize was announced. And Rick McCann, who was in that lab at the time, has said that they couldn't even get funding. After they cloned GFP, they still couldn't get funding for it because people just didn't realize how earth-shaking it was. But I also think that you shouldn't, now it's ubiquitous. It's used in all kinds of biomedical situations. It's used to help cure cancer. There's so many applications, but the motivation really was basic research and to figure out how does this jelly make light? What's the mechanism? And so it wasn't that long-term vision of, wow, we can make millions of dollars if we can get something that glows when you put the genes in. So I think this is kind of a sad story about science, but hopefully it's also an inspiration that we should help basic research and exploration. So speaking of applications, the GFP and later red and yellow and orange fluorescent proteins that were cloned from corals, they're really useful so you can label live organisms and look at them under fluorescence without having to cut them open at times. Bioluminescent chemicals like Aquarium and other luciferases are also extremely useful. They don't require you to shine light through the tissue and have the light come back out. They are emitting light from inside the tissue, so they're actually more sensitive. They were originally used as calcium indicators to look at things like fertilization of eggs and this calcium wave that goes around, how muscles contract with calcium. But there's just all kinds of applications for these biological highlighters. I even like to think that this Nobel Prize in 2014 is somewhat related to fluorescent proteins because it's an imaging method that gives you super high resolution, and it's based on, I think, the lifetime of fluorescence in some of these fluorescent molecules. So that was like somewhat Dr. Shimomura's work and the ramifications for that. And now I want to talk a little bit about where we're going with this and some of the research that we're doing with fluorescence and bioluminescence both. So for fluorescence, this is a deep sea anthozoan, like it's called an octocoral sea pansy kind of thing. I don't know what you call a sea pen. So this is the white light view of it. If you look at it with fluorescent light, looks totally different. This one also happens to be bioluminescent and fluorescent. But so how does fluorescence work again? So you have a molecule, some kind of light packet of energy comes in and excites an electron in that molecule to a higher energy, but it's not stable. It only temporarily lives at that excited higher energy state. Here you can see the energy kind of decaying down and then it drops down. And this actually happens almost instantaneously. I've just slowed it down for this animation. When the energy comes back out, you get another photon back out, but that photon has less energy than what went in because it's not a purely efficient process. So you have higher excitation energy, and this is why people always talk about UV light for excitation. It doesn't have to be UV, actually. Blue light is more efficient in many cases. But then the longer wavelengths are the ones that are coming out that are the fluorescence excitation. And again, you only get light out as long as you're shining light on there. So to take the pictures that I'm going to show you, what we do is we put the blue light on, but then we put a yellow filter, and that blocks the blue light and lets those longer wavelengths come through so we can take them. So in the environment, if you look at something, it's very drab-looking brown coral, right? But then when you shine blue light on it, suddenly it looks totally different, right? Because it's got all these fluorescent proteins inside of its tissue. Similar for just an intertidal shot of, here's a sea anemone with some anemones along the rocks, and then the blue light view of it is totally different. We have these yellow fluorescent proteins from the strawberry anemones, green in this anemone, and then a lot of this red is algae and other anthozoans. So this is one of the ones that we worked on to clone the fluorescent proteins. This is the white light view and the blue light view again. This one has 7 different genes for different colors of fluorescent proteins that can be in a single polyp. And you can take those genes out, and this is what makes GFP so amazing, is that you can take that strip of DNA out and put it into bacteria. It'll fold itself up into a little barrel and automatically activate the chromophore. And so you can grow it into bacteria and get these test tubes. And I have some test tubes that we cloned like 10 years ago, and they're just on the outside of our fridge, and they're still fluorescent. So you can imagine a protein like, put a vial of milk outside your refrigerator for 10 years and see what it looks like. But these are incredibly durable and useful proteins. So to kind of get an idea of how these fluorescent proteins are distributed in the water, we put a blue light on our ROVs, and then we can fly around with blue light. We can have the white lights on and then find an organism, turn the lights off, and you can see just that fluorescent ring illuminated around the margin. This is a species that we described recently with the white light. You can already see the fluorescence there, but there with blue light, just incredibly bright fluorescence. So those guys are both bioluminescent organisms. So we know how they get the energy that is used with the fluorescence, but there's actually a whole bunch of organisms out there which are fluorescent, but they're not bioluminescent. And one of them is this flower hat jellyfish. And under blue light, it's got these tentacles that have this kind of incredible fluorescent tip to them. They also actually have a pink tip, which is called a chromophore, so it absorbs blue light but doesn't re-emit the fluorescence. So it's got a really extra dark spot right next to an extra bright spot. So we had some suspicions about what it was doing with this, set up some experiments in a tank where we could change the lighting regime and then have a fish, and we would kind of like release the bowl into the bowl ring, but we could raise this door and show the fish the jelly under these different lighting things. So we tried white light, we tried the yellow light that was too long to stimulate the fluorescence, and then we would try the blue light. And when we did the blue light, fish suddenly became very interested and would actually attack the glass trying to get at those fluorescent tentacle tips, as I'll show you. And this happened at a really significantly different level. So what we figured is that the jelly is using that fluorescence to actually attract prey. It's like a supernormal stimulus, it's called, where you have something that's an exaggerated signal where the fish is looking for any little thing that it can possibly pounce on. And when it sees this green light, it goes right for it. So we made an underwater laser pointer, a green underwater laser pointer, and we took it to the barrier reef and shined it around on the barrier reef. And we got pretty good evidence that this phenomenon can happen in nature. So, yeah. The fish would get really annoyed that they could not catch that little green bug that was swimming around. So you can imagine if you were trying to catch fish, and actually I was on the boat, there was somebody who was studying fish feeding responses on the boat at the same time, and he would spend hours underwater waiting for the fish to do one little jaw thing, and then it was like, saw this, and he was like, oh boy, I've been doing my... Once we saw that in this jelly, it kind of opened my eyes to a lot of other places where this same phenomenon is probably going on. So this is a tube anemone, has fluorescence localized in a very suspicious point, in its body. There's also a siphonophore, which feeds on fish. And there was a paper about how it's strange that this only feeds on larval fish only during the day and not at night. And they couldn't figure out why, because there were larval fish both places. And when I looked under the fluorescence conditions, each one of its tentacles has a little fluorescent dually bopper on it. And so those are going to be really apparent to the fish under dim blue light, but obviously not at night. So we think that this is actually somewhat widespread phenomenon. Now, there are a lot of organisms that look pretty spectacularly fluorescent, but it's not clear whether or not there's actually a function to it. So this is a shrimp. It happens to have fluorescent banding on it. And you might hear about the biofluorescent turtles or biofluorescent puffins. I like to present the world's first biofluorescent human. So a lot of our structures in our own bodies are also fluorescent. So your fingernails, the lens of your eye, and then the red fluorescence in a lot of those things is from chlorophyll from the plants. So just not all that glitters is gold necessarily. There's a lot of fluorescence. Cheez-Its are fluorescent, for example. There's a lot of fluorescence just out in the environment, and it's not necessarily related to their function. All right, so the last thing I want to talk about is bioluminescence and some of the recent findings about bioluminescence. If you run a trawl net through the water, like a big fishing net, you collect all the organisms into a reservoir at the end of it. That's what we've done here. And then this is a big tank, like a small bathtub or a large tank, depending on if you're an optimist or a pessimist, that we set up on the ship to empty the samples into. And so if you do that with the lights off, you can see just how many things in that plankton net are bioluminescent. You can see the different colors of bioluminescence. There's blue and green in here. So with my postdoc Severine Martini, we looked at our old ROV video records, and we cataloged all the organisms that were in there, and then we made a database of bioluminescence capability, of definitely bioluminescent, likely, unlikely, or not bioluminescent. And when we kind of crossed those two databases, we found that 3/4 of the organisms that we could see from the ROV were bioluminescent. So throughout the water column, all the way down to 4,000 meters. And who it was at different depths changed, but overall it was a big diversity. And then some groups would be like really bioluminescent, like the siphonophores and ctenophores, and some groups would hardly be bioluminescent at all, like the salps. And in this list, it's far from comprehensive, but all the red ones are ones where we don't know all that we could know about the chemistry. So these are some of the targets for research now, is just the unknown chemistries. A lot of them do use cylindrazine, as I'll show you in a minute. So another way to look at this diversity is with the tree of life view. And this just goes from bacteria on one end to starfish on the other, so spanning the range of life. And if you look at it in terms of luciferins, so the light-emitting molecules, these are all the ones that use bacteria. I think a lot of people think that bioluminescence is largely bacterial, but it's actually really pretty uncommon. These are ones that use sort of special luciferins, and that orange dot there is the one that Dr. Shimomura first crystallized. That's from those ostracods here. And then these are all the ones that use cylindrazine. So of course you know which is my favorite molecule on Earth is cylindrazine, which he also figured out and characterized. And this is just found across the tree of life. These are some of the unknowns. Again, there's quite a few for you young scientists out there. There's quite a few unknowns still in the world. Not everything is known. These are the things that have green fluorescent protein. And then these are all the things that Dr. Shimomura worked on or contributed to. So he really just hit the range of organisms and made profound contributions across the tree of life. So I just showed you that diversity. What's a little bit strange about it is that the number of light emitters, the luciferins that are used, is actually really small. There's just a handful that we know of in the ocean. So these are the big ones. As I showed you, cylindrazine is used for a huge diversity of organisms, everything from single-celled organisms up to, again, we think it might be in some echinoderms, some starfish, and then I'll show you some more examples with these. And then these are some of the kind of exciting ones that are the unknowns, where we just don't know what their light emitter is yet, or other molecules. So all that cylindrazine is out there. How do the jellies, like who is making the cylindrazine? We know the genes for photoproteins, but one of the basic questions is who is making the cylindrazine itself? And so we did this experiment, again, using Aquaria, so this is like a known entity, but it had been raised down in the aquarium for many generations. And we checked it, and it actually had lost its ability to bioluminesce through time. So this was really suspicious, and we figured that it can't actually make cylindrazine, and in the wild, it's probably getting it from something that it ate. Aquaria actually eats both jellies and other kinds of plankton. So we took Aquaria that had never been dark, even their grandparents, had never made light. Even their grandparents were dark, it's like they had never, ever, ever made light. And we injected cylindrazine in them, or we fed them with prey that had cylindrazine, or we put cylindrazine in the water, and they immediately became able to make light. So they were making the photoprotein part of it, but they didn't have the batteries to put in the flashlight. And again, there was a thumbprint on the previous slide, but this was cylindrazine that was provided again by Dr. Shimomura that enabled these experiments. So what we inferred from that is that actually something else upstream is making the cylindrazine that the jellies, that are so famous that it's named after, that it came from. So since this, we're trying to figure out how it's synthesized, and there's been this hypothesis in the literature that it's made from FYY, so phenylalanine, tyrosine. These are 3 amino acids, so like 3 little balls, and that these chemicals can come together and be transformed into cylindrazine. And there were some really experiments by Dr. Oba and Inoue and colleagues where they did isotopically labeled amino acids so they could know if they were incorporated into the cylindrazine that came out. And in these copepods, they showed that it was actually synthesizing cylindrazine from FYY. So here again, it's the mechanism. You have these 3 amino acids. And what what you really need is something that can form this oxygen bond right here. That's called the cyclization step. So there's two kind of prerequisites for this. And so with Warren Francis and Nathan Shaner, he was actually a grad student in Roger Chin's lab who got the Nobel Prize for GFP. He did a postdoc in my lab, but it's all really kind of interconnected when you get into bioluminescence. So did some experiments to try to look for enzymes that would have these properties. And as we were looking through these transcriptomes, which is like a catalog of all the genes sequenced, we noticed something a little bit suspicious, which is that there was this one gene that ended in FYY, okay? So that's like, well, red flag going off. And when you looked at the rest of the gene, it actually is something that is involved in that cyclization process. So we think it's like when you buy the razor and it comes with the blades, maybe this is the protein and the thing that it's gonna act on are all tied together in one thing. One other piece of evidence is that FYY gene was only in the bioluminescent species of ctenophores, and there's two non-bioluminescent species that we looked at their genes, and they didn't have that gene to make, that we think is involved in slinerzine. So it's all circumstantial evidence, but it's we think pointing us in the right direction that gene may have something to do with GFP. And so right now we're trying to actually knock out that gene in an animal and see if it goes dark because it doesn't have the proper slinerzine. All right, so let's talk a little bit about why is bioluminescence so widespread across all these ecosystems. I think part of it is just the principles of communication. So if you imagine there's an organism that's a millimeter across and it's, say, just at the other side of that stage, and it wants to communicate to me. What are the options that it has to communicate over that distance? It can't use sound, right? It can't use a chemical cue because it would take so long to get over here. So really, a flash of light is the only way that something of that scale could efficiently communicate across such distances. And this is like equivalent to somebody standing on a mountaintop. 4 kilometers away or 20 kilometers away on a 4-kilometer mountain. If you're gonna talk to that person, you're gonna have to use a signal mirror or something, right? You can't communicate by any other means. So organisms can use this to amplify and to communicate a powerful signal when they might be operating at a really different scale. A lot of the functions that we hypothesize for bioluminescence are defensive, so either hiding yourself, scaring your predator away, and if you want, if you go to the bioluminescence webpage, which I'll send the link at the end, it has more of an explanation of all these. These are just the cartoon version of it, and also in that review paper. But the flip side of that is the offensive functions that we often think about, like an anglerfish luring prey. Some actually use them as little bioluminescent searchlights to find prey, so they'll have a spotlight or a red spotlight even. And then there's actually very few examples that we know of where they use it to communicate for mating purposes, but that's partly because it's a hard thing to study in the ocean. So with this concept of using it as a lure, here's an example of a somewhat unusual anglerfish that we found. But what I think even more unusual is that we found these siphonophores. There's 5 species of this siphonophore that all eat fish, and they have at the tip of their tentacle, right where all their stinging cells are, they have this little transparent stalk with like a Tootsie Pop at the end of that. And that Tootsie Pop, in the inside, it has bioluminescent material, and the coating is a fluorescent, this red fluorescent material. So they flick them when they are exposed to light. And so again, we think it's jigging for its fish prey. Instead of spreading out its tentacles really broadly and just trying to cast a net, like some of the ones I showed you earlier, this is actually an angler siphonophore that's operating in the deep sea where fish are few and far between. Another pretty interesting one is the vampire squid. And this one has bioluminescence in its arm tips. And what's more is that if it's in good condition and if this was with your eye instead of a low light camera, it actually exudes out those particles. So these are the particles at the tips of those arms. And we found also that this uses cylindrazine using the chemicals that Dr. Shimomura provided me with. I was able to show that this vampire squid also uses cylindrazine for its bioluminescence. And so right now we're trying to clone the luciferase that it uses to cross-react with that luciferin. Like milking the tentacles of a vampire squid, which is not something everybody gets to do. Another one that we're working on is this polychaete worm called Tomopteris. It can be up to like a foot long, but it's unique in that it makes yellow light. Everything else that I've shown you has made blue or green light, right? This is one of the few things in the ocean that makes yellow light. And my student Warren Francis actually figured out the chemical structure of that yellow fluorescent material. So we don't know for sure whether it's the luciferin or not, but we know it's somehow involved in the bioluminescence, is that chemical. There are also a lot of polychaete worms. This is one that we found that has, we call it the green bomber. It has these little sort of bombs next to its neck, and it sheds those off into the water. And again, those are filled with bioluminescent material. So we can take one of those, and we don't have to necessarily cut up 50,000, to try to purify it out of the whole tissue. We can just get that's pre-purified for us. So the last example that I want to talk about was something that I did. I wanted to acknowledge my advisor, Jim Case. He worked originally on fireflies a lot in Indonesia and around, and came to marine bioluminescence later because it actually paid the bills because the Navy was interested in bioluminescence. But he loved technology, he loved bioluminescence. The cameras that we used in the lab at the time were these intensified low light cameras. So it's like a really grainy old television that you're trying to tune in to the station. So this is a pretty neat experiment that was done in the lab with demonstrating counter illumination, but I'm showing it to you here just to show the kind of video that we were able to do at the time. I think it's like 480 pixels by 340 or something, and black and white. And since then, with some engineers at Mbari, we've put low light cameras on the submersible so that we can take them down and actually look at bioluminescence in full color in the environment. So just as an example of that, this is a sea cucumber. Looks pretty boring. These are just covering the abyssal plain, but I think this is about 2,500 meters deep, so pretty deep. But if we take the manipulator arm, we take one of those, hold it in front of our camera, then we get to see what its bioluminescence really looks like. And again, this is really hard. If you take one of these cucumbers up to the surface by any normal means, it probably doesn't even have any bioluminescence anymore because it's just been exhausted. But this is an example of what we can see. So I was really fortunate in grad school to be invited to a lot of cruises with Edie Witter. And on these, we did blue water diving, we did submarine dives. Peter Herring also would come along, and he's sort of like the one who at the time just knew everything about what organisms were bioluminescent or not. And so we would go down in these subs and look out the window and, see all the bioluminescence happening in the environment outside of our window. And on one of those dives, I think it was on my birthday actually, but I was looking out the window and I saw one of these little inch-long arrow worms that darted away. And you can't see it here really, but there's like a cloud of yellow particles that comes out behind it. When I could see it with my eye, I could see that it shot out bioluminescent material. But these arrow worms were not supposed to be bioluminescent. So I was on a quest to find out what it was that I saw, and it took me like 3 years to figure out that it was this arrow worm. They're pretty common in the plankton. They use these hooks to catch their prey. But in this particular case, it had bioluminescent organs that were on the lateral sides of the fins, and when it undulated its body, it would shed off those particles into the water. So again, guess which molecule this uses to emit light? Cylindrazine, yeah. So I was able to use the chemicals that Dr. Shimomura had given me to show that this used cylindrazine as well. But this was like my first Nature paper as a grad student. I was really excited about this discovery. Also, this was film. This was before digital cameras. So I was able to get the bioluminescence and the thing, and this is the kind of thing you'd go out to sea for 3 weeks, take some pictures. You'd have no idea if the exposure was over, under, did it even show up until you got back and, developed the film a week later. So this was really lucky that I got that bioluminescence to show up. So, of course, the ocean is full of surprises, and as we went out and looked, the other orange keyed gnat that I had kind of ruled out in my study turns out to have a bioluminescent organ, but in this case it's on the tip of its tail and not on the side of its body. So these are two unrelated organisms that have bioluminescence, evolved apparently somewhat independently. These are the structures of their bioluminescent organs, so it's just chock full of these grains of bioluminescent material, and this is the tail organ. So my student, Darren Schultz, we looked through the transcriptomes, and we think we have the luciferases for both of these keyed gnats, which is kind of an exciting full circle kind of thing. The other full circle is that we have our ROV with a low light camera mounted on it, right? So one of the first targets was to go down with our camera and find one of these keyed gnats underwater so I could film what I had seen from the sub like 25 pounds ago when I was a grad student. So again, here's one of these worms that's hovering in front of our camera, and as we sit, it does its escape response and shoots out that burst of things, which is exactly what I saw out the sub window. So technology is, it's enabling us to make some discoveries that I think both my advisor, Jim Case, and Dr. Shimomura would have appreciated, and we're still trying to build on the shoulders of this really, monumental man, and I'm really appreciative of him for not just his scientific contributions, but his contributions to my development as a person and a scientist. There's more information here. I didn't have time to talk about Jelly Watch. That's a separate citizen science project, and I do have stickers for that one. And then also a lot of this information is on our bioluminescence webpage or in the publications. So if there's time, I'll take some questions. Thank you. I guess, so if you have a question, I guess take a microphone.
Yep. So we have time for just a couple of questions, and just one reminder, there is a reception in the Meg's room in the Swope Center following where you can continue the conversation with our speaker. And there is a slideshow of Dr. Shimomura's life. So some really significant photographs from his life. Just a couple of questions. One on your right, Dan.
Thank you. That was fascinating. You had said that some of the organisms are making their blue light in order to fluoresce the green light. How many of those organisms make the blue light to fluoresce green light in other organisms?
In other organisms? Like to shine it around and find fluorescence of another organism? Probably not a lot. The fluorescence that happens in the cells themselves, it doesn't actually even come out as light. They're so closely associated that the energy kind of goes straight to the other protein. So it'd be kind of inefficient to do it that way, but there are ones that use searchlights to find their prey, and there could be some fluorescence component to that. That's an interesting thing I hadn't thought about.
So that was a really great talk. I was wondering, in these fish that seem to be attracted to the green lights, what is the evolutionary advantage of that sort of insane attraction to something that in some cases is their death?
Again, there's an ecological concept called the supernormal stimulus. So a seagull will sit on an egg that's larger than a normal egg because it's like over-triggering their egg pathway. So they'll sit on an egg and neglect their real egg because this one's larger. And a baby seagull will go to like a giant red spot on the beak of a seagull as opposed to a realistic spot on a seagull. People will eat like a Big Mac instead of an actual hamburger because it's like a super exaggerated version of actual food. So they're I think hardwired to say, anything that is bright and moves is likely to be, something that delicious, yeah. And evolutionarily that plays out to their advantage over long term, but then things have kind of turned it on its head.
Okay, well, let's thank Dr. Haddock one last time.
Why do we assume it's light of blue light if there isn't a source of blue light? What do you think the reason for that?
The light that filters down through the ocean is predominantly blue, so the eyes of the organisms that are living there are adapted to see blue. Some of them only have one pigment, and it's a blue pigment. So it's kind of an interplay between the visual sensitivity, the environment itself, and the colors that transmit best in the environment, and then the chemistry.
But then we're not seeing it bioluminescent.